Hello everybody, I'm Nasreen. I'm a PhD student from Cranfield University and I'm here to present my work that I presented at the IWA AD18 conference in Istanbul, Turkey in the beginning of July. And I'm going to present on enhancing anaerobic digesters efficiency through thermal pretreatment, and I'm going to talk on the impacts of sludge composition and trace element bioavailability in full-scale sites. My PhD work is sponsored by Anglian Water, a leading water and wastewater utility in the UK. At present, Anglian Water employs three different kinds of pretreatments in their site, namely thermal hydrolysis, enhanced enzymatic hydrolysis, and heating pasteurization and hydrolysis, with the latter two being biological hydrolysis. And these pretreatments have been employed to engineer out the hydrolysis step, which is the rate-limiting step in anaerobic digestion. However, there is very little information on the effect of pretreatments and the different types of pretreatments on the composition of the feed. And at the moment, optimization is defined by feedstock rather than the composition of the feedstock, and more importantly, their fate after pretreatment. What we also don't know is whether optimization has been fully achieved, or if there is a gap in optimization which could be further enhanced by the addition of hydrolytic enzymes or limiting trace elements. And for today's presentation, I'm going to talk about my work on the thermal hydrolysis site. To investigate the effect of pretreatment on the composition of digestive feed and digestate, I characterized the sludge from full-scale sites, particularly looking at macromolecule and trace element composition. The aim of this is to determine whether pretreatments have a bias towards certain macromolecules and whether macromolecules are only partially hydrolyzed in some pretreatments. I then take my study to the lab scale, where I investigated whether the addition of hydrolysis enzymes enhances the biomethane yield of pretreated sludge by potentially targeting the partially hydrolyzed macromolecules. This was done through carrying out batch biomethane potential test. I'm currently working on assessing the long-term effects of enzyme addition into pilot-scale continuous digesters receiving pre-treated sludge using enzymes pre-selected from the batch tests. I'm also looking at the effects of trace element addition, whose selection is done through prior characterization studies. Ultimately, the study is aimed to inform optimization at full scale. For this presentation, I'm going to discuss the results that I gathered from the full-scale sludge characterization and the BMP tests with hydrolysis enzymes. For my sludge characterization study, I looked at two different sites. The first site is the site that employs THP pretreatment. It is a large-scale site, and the THP pretreatment occurs at 165 degrees Celsius and 6 bars for 30 minutes. In order to compare the results of pretreatment with one without, I'm also looking at a sewage treatment site that does not have pretreatment, and it's a medium scale site. In this slide, I'm going to talk about the results of the macromolecule characterization study I did for both the THP and the conventional site. The biggest impact of THP pretreatment is on the solubilization of proteins at around 30%, with no further solubilization after digestion, and the solubilization of carbohydrates by 27% after pretreatment. But the biggest impact is the solubilization of carbohydrates after digestion, which amounted to close to 86%, compared to the conventional AD or conventional digester, which only achieved around 30%. So what this shows is a bias in thermal hydrolysis pretreatment towards the solubilization of proteins, which is a greater bias, and the greatest bias on the solubilization of carbohydrates both after pretreatment and after digestion. I also looked at the soluble profiles at both the THP and conventional sites, where I investigated the presence of soluble COD, VFA, total ammonia nitrogen or TAN, and the ratio of VFA to soluble COD. In the pre-treatment side, it was before pre-treatment, after pre-treatment, and after digestion. And in the conventional side, it was before and after digestion. So we see that pre-treatment doubles the concentration of soluble COD while having not much impact on the increase in BFA. However, we see that there is a much greater presence of residual soluble COD in the digestate receiving THP pre-treated sludge compared to a conventional digester. This is also indicated by the low VFA to SCOD ratio in the THP digestate as compared to that in the conventional digester. And this suggests that not all of the solubilized macromolecules have been converted into VFA, indicating that enhanced solubilization achieved by THP does not necessarily lead to the enhanced processes or reactions that are occurring after the hydrolysis or the solubilization step. I'm now going to discuss the impact of thermal hydrolysis on trace element bioavailability. Trace elements have an important role to play as enzyme cofactors in various stages of anaerobic digestion. However, their total concentration does not directly relate to bioavailability. So in my study, I classified the trace elements into 5 different fractions, with the first 2 being bioavailable and fractions F3, F4, and F5 being less bioavailable as we go down the hierarchy of fractions. So these are heat maps of trace element speciation and mobility for the THP side. This is for before pretreatment, after pretreatment, and after digestion. And for conventional side, this is for before digestion and after digestion. We see that most of the trace elements are concentrated in more stable fractions in both the sites. However, THP pretreatment increases the bioavailability, or it releases the stable fractions into more bioavailable fractions, especially in the case of selenium. After digestion, we notice that there's a greater mobilization of trace elements towards back to the stable fractions in the digester receiving THP pre-treated sludge, as compared to one that does as compared to a conventional digester that does not have pre-treatment, suggesting that THP pre-treatment it encourages the mobilization of trace elements into more stable fractions, even though they manage to increase the bioavailable fractions of some trace elements like selenium after pretreatment, suggesting that there could be a greater demand of trace element or greater trace element efficiency in THP digesters as compared to a conventional digester. So after having investigated the effect of pretreatment on sludge composition at full scale, I then took my study at lab scale, where I investigated the effects of adding hydrolysis enzymes on the BMP, on enhancing the BMP of the THP sludge. And we see that the enzymes that targeted proteins, namely protease and alkylase, had the greatest impact on enhancing the BMPs at different concentrations, starting from 0.5 to 4% of the enzyme with respect to the total solids of the sludge. And this is consistent with the observations of increased solubilization after THP pretreatment, especially for proteins, and their less efficient utilization in the digester with regards to their conversion to VFA. So we see that the addition of these enzymes help bridge that gap where you see an increased solubilization of proteins, but these proteins are then are broken down by the enzymes, which are not generally broken down in the digester itself, and that has contributed to the enhanced BMP as we observe here. So in conclusion, I'd like to leave you with 3 main takeaways. The first is that solubilization does not necessarily translate to utilization in anaerobic digesters. The second is that THP pretreatment mobilizes the trace elements into more stable fractions, indicating greater TE requirement in digesters receiving THP pretreated sludge as compared to conventional digesters that directly receive the sludge. And the last point is that THP achieves partial hydrolysis of proteins, and adding protein hydrolyzing enzymes can enhance the BMP of digesters receiving THP pretreated sludge. So the way forward for me is I'm looking at long-term tests with pilot scale continuous ADs, independently studying the enzyme and the effects of enzyme and trace element dosing in the long term. And I'm also looking at the synergistic supplementation of enzymes and trace elements. And ultimately, all of this is to inform full-scale optimization for both for all the sites in that incorporates both thermal and biological pretreatment. So this is the end of my presentation, and I would like to firstly thank EBNet for funding my conference, and it was an amazing experience, which could not have been possible without this funding. I'm also grateful to the exceptional guidance and support from all the three of my supervisors, Dr. Yadira and Dr. Frederick from Cranfield University and Julia from Anglian Water. I'm also grateful to my summer interns, Arizu and Subhashini, who were of great help for me and without whom I'd just be talking to my reactors in the lab. So thank you very much. And if you have any questions, please feel free to reach out to me on my email.